The human brain is a high-demand organ, consuming roughly 20% of the body's energy despite weighing only about 2% of total body mass. Much of this energy supports neurotransmitter synthesis, synaptic signaling, and maintenance of electrochemical gradients. Among the various energy sources, glucose is paramount, but it's not always immediately available in the quantities neurons require, especially during periods of stress, fasting, or high cognitive load. This is where brain glycogen, largely stored in astrocytes, enters the picture. Though once thought to be metabolically insignificant, glycogen and its shuttling mechanisms now appear to play a key role in sustaining neuronal function and, more intriguingly, in regulating emotional states and mental health. Glycogen shuttling refers to the process by which astrocytes mobilize glycogen stores and deliver metabolic support to neurons. During periods of increased demand, such as learning, emotional arousal, or exposure to psychological stress, astrocytic glycogen is broken down into lactate, which is then transported to neurons via monocarboxylate transporters. This lactate is not just a passive fuel substitute. It can act as a signaling molecule, modulating neuronal excitability, plasticity, and neurotransmitter dynamics. The astrocyte neuron lactate shuttle is increasingly recognized as a critical component of the brain's adaptive response to stress and may influence behaviors tied to mood, motivation, and resilience. The synthesis of mood-regulating neurotransmitters such as serotonin, dopamine, and gamma-aminobutyric acid, GABA, is highly energy-dependent. Each step of their biosynthetic pathways requires ADP, cofactors, and tightly regulated enzyme activity, processes that falter when energy availability is unstable. If glycogen stores are depleted or astrocytic shuttling is impaired, neurons may experience subtle but chronic energy deficits. Over time, this could dampen neurotransmitter production, reduce synaptic strength, and impair the regulatory circuits involved in emotional balance. In fact, this kind of neuroenergetic insufficiency is being explored as a unifying model in mood disorders, particularly depression and anxiety. Animal studies have demonstrated that stress-related elevations in corticosterone reduce brain glycogen levels, particularly in limbic regions like the hippocampus and prefrontal cortex, areas heavily involved in mood regulation. Chronic stress may thus impair glycogen availability at precisely the moments when emotional resilience is most needed. Conversely, behavioral interventions known to improve mood, such as exercise, sleep, and cognitive engagement, also enhance glycogen turnover and lactate transport, suggesting that healthy glycogen cycling may be one of the hidden engines of emotional recovery. Moreover, disorders such as major depressive disorder, MDD, and bipolar disorder have been associated with abnormalities in glial cell function, especially astrocytes. If astrocytes fail to supply adequate metabolic support due to impaired glycogen storage or defective lactate shuttling, the consequences may go beyond mere energy shortage. Neuronal firing patterns become erratic, long-term potentiation may be blunted, and the prefrontal amygdala circuitry that governs fear, impulse control, and mood regulation may fall out of sync. This could help explain why many individuals with mood disorders experience cognitive fog, emotional dysregulation, and poor stress tolerance even when their primary symptoms appear controlled. There's also growing interest in how sleep, glycogen, and mood are intertwined. Glycogen levels in the brain fluctuate with the sleep-wake cycle, depleting during wakefulness and rebuilding during deep sleep. If this cycle is disrupted, such as an insomnia or circadian misalignment, glycogen restoration may be incomplete, leading to chronic energy deficits in emotionally salient brain regions. Poor sleep is both a symptom and a driver of many psychiatric conditions, and disrupted glycogen dynamics may be a contributing link in this vicious cycle. Importantly, lactate derived from astrocytic glycogen may have direct neuromodulatory effects. Studies have shown that lactate can increase expression of brain-derived neurotrophic factor, BDNF, a molecule critical for neuroplasticity, learning, and mood regulation. BDNF levels are typically reduced in individuals with depression, and interventions that elevate lactate, such as high-intensity exercise or caloric restriction, also tend to raise BDNF. This hints at a mechanistic bridge between glycogen metabolism and emotional resilience. The lactate shuttle isn't just a biochemical workaround for glucose shortages. It's a dynamic communication pathway that may shape how we experience and recover from emotional events. Despite these promising connections, several caveats remain. Most human research on glycogen and mental health is indirect or inferential, often extrapolated from imaging studies or cerebrospinal fluid analyses. We still lack precise tools to measure glycogen dynamics in the living human brain in real time. And while experimental models have shown strong associations between astrocyte metabolism and behavior, the translation into clinical psychiatry is still at a nascent stage. Still, the implications are powerful. If emotional regulation is partly governed by the health and responsiveness of astrocyte neuron energy networks, then therapies that target glycogen dynamics may offer a novel path forward. This might include pharmacologic agents that enhance glycogen mobilization, dietary strategies that stabilize glucose availability, or behavioral interventions that improve lactate signaling. Such approaches could complement traditional psychiatric medications, especially in individuals whose symptoms are intertwined with fatigue, brain fog, or stress sensitivity. In conclusion, the humble glycogen granule, once dismissed as irrelevant in the adult brain, may in fact hold vital clues to how we manage stress, regulate emotion, and maintain mental stability. Through its influence on neurotransmitter synthesis, neuroplasticity, and sleep-dependent restoration, glycogen acts as both an energy reservoir and a signaling nexus. When this system is functioning optimally, it supports the brain's emotional equilibrium. But when disrupted, it may contribute to the subtle metabolic imbalances that underlie modern epidemics of mood disorders. As neuroscience continues to probe deeper into the energetic roots of emotion, glycogen shuttling may emerge not just as a supporting actor, but as a central player in the drama of mental health.